Hi class, this is your instructor, Skylar Huff. We're now in chapter 20, and this is Speciation and Macroevolution. So as you can see there, over evolutionary time, various environments on Earth abound with rich assemblages of species, and scientists about now have named nearly 2 million of the approximate 4 to 100 million existing species. But given that, you should also take into account that more than 99.9% of all species that have ever existed are now extinct. I guess I should say thanks a lot, people on Earth. So what you're seeing right there before yourselves are blue-footed boobies. So they are a large seabird that nest on the Galapagos Islands and several other islands in the eastern Pacific Ocean. Male and female pairs are monogamous and typically nest on black lava. The largest blue-footed booby breeding populations are found on the Galapagos Islands. So let's get into what is a species. The concept of distinct organisms, known as species, is not new. However, there are several different definitions of species, and each definition has a sort of limitation. So Linnaeus, who founded modern taxonomy, the science of naming and describing and classifying organisms, he named and classified plants and other organisms into separate species based on their visible structural differences, such as feathers or the number of flower parts. This method is known as the morphological species concept, and it is still used to help characterize species. But structural differences alone are not adequate to explain what constitutes a species. So here nextly, population genetics did much to clarify the concept of species, according to the biological species concept, and this was expressed by Mayer. A species consists of one or more populations whose members interbreed in nature to produce fertile offspring and do not interbreed with, that is, are reproductively isolated from members of different species. So individuals belonging to the same species tend to share common features because their genes are derived from a common gene pool. So gene flow occurs between individuals of a given species when they mate, but reproductive barriers restrict individuals of one species from interbreeding or exchanging genes with members of other species. As a result, reproductive barriers keep a given species genetically distinct from other species. An extension of the biological species concept holds that a new species evolve when formerly interbreeding populations become reproductively isolated from one another. So the biological species concept, it is the most widely accepted definition of a species, but it has several shortcomings. So it applies to those species which are only able to reproduce sexually. It does not apply to bacteria and other asexually reproducing organisms, or to extinct organisms which are classified by structural or biochemical characteristics. And lastly, individuals assigned to different species, such as coyotes and wolves, sometimes do successfully interbreed. So you're seeing here a rotifer. So this rotifer, these deltoid rotifers, are all females. And because they do not reproduce sexually, it is difficult to classify these small marine animals as species under the biological species concept. The phylogenetic species concept. So some species prefer this concept, also called the evolutionary species concept, in which a population is declared a separate species if it has undergone evolution long enough for statistically significant differences in diagnostic traits to emerge. So if a population has a unique diagnostic trait, a set of spines running down the back, for example, it can be demonstrated to be inherited. This population is considered a separate species. The shortcomings state that it requires thorough studies of DNA sequences, and many subspecies will become separate species. Now let's get to reproductive isolation. So various reproductive isolating mechanisms prevent interbreeding between two different species whose ranges overlap. These mechanisms preserve the genetic integrity of each species because gene flow between the two species is prevented. Most species have two or more mechanisms that block the chance of occurrence of individuals from two closely related species overcoming a single reproductive isolating mechanism. Although most mechanisms operate before mating or fertilization occurs, which is termed prezygotic, others work after fertilization has taken place, which is termed postzygotic. Prezygotic barriers are reproductive isolating mechanisms that prevent fertilization from taking place. Because male and female gametes never come in contact, an interspecific zygote, that is, a fertilized egg from the union of an egg from one species and sperm from another species, is never produced. Prezygotic barriers include temporal isolation, habitat isolation, behavioral isolation, mechanical isolation, and gametic isolation. Sometimes genetic exchange between two groups is prevented because they reproduce at different times of the day, season, or year. Such examples demonstrate temporal isolation. For example, two similar species of fruit flies, known as Drosophila, have ranges that overlap to a great extent, but they do not interbreed because one species is sexually active in the afternoon only, and the other is sexually active only in the morning. Similarly, two frog species, as you see here, have overlapping ranges as well, and this is in eastern Canada and in the United States. The wood frog usually mates in late March or early May when the water temperature is about 45 degrees, whereas the northern leopard frog usually mates in mid-April when the water temperature is about 55 degrees Fahrenheit. Although two closely related species may be found in the same geographic area, they usually live and breed in different habitats in that area, providing habitat isolation between the two species. For example, 5 species of small birds known as flycatchers are nearly identical in appearance and have overlapping ranges in the eastern part of North America. They exhibit habitat isolation because during the breeding season, each species stays in a particular habitat within its range, so potential mates from different species do not meet. The least flycatcher frequents open woods, farms, and orchards, whereas the Acadian flycatcher is found in deciduous forests, particularly in beech trees and swampy woods. The alder flycatcher prefers wet thickets of alders. The yellow-bellied flycatcher nests in conifer woods, and the willow flycatcher frequents brushy pastures and willow thickets. Many animals exchange a distinct series of signals before mating. Such courtship behaviors illustrate behavioral isolation, or also known as sexual isolation. Bowerbirds, as you see here, exhibit species-specific courtship patterns. The male satin bowerbird of Australia constructs an elaborate bower of twigs, adding distinctive blue parrot feathers with white flowers at the entrance. When a female approaches the bower, the male dances around her, holding a particularly eye-catching decoration in his beak. While dancing, he puffs his feathers, extends his wings, and sings a courtship song that consists of a variety of sounds, including loud buzzes and hoots. These specific courtship behaviors keep closely related bird species reproductively isolated from the satin bowerbird. If a male and a female of two different bowerbirds begin courtship, it stops when one member does not recognize or respond to the signals from another. Sometimes members of different species court and even attempt copulation, but the incompatible structures of the genital organs prevent successful mating. Structural differences that inhibit mating between species produce mechanical isolation. For example, many flowering plant species have physical differences in their flower parts that help them maintain their reproductive isolation from one another. In such plants, the flower parts are adapted for specific insect pollinators. 2 species of sage, for example, have overlapping ranges in Southern California. Black sage is which is pollinated by small bees, has a floral structure different from that of white sage, which is pollinated by large carpenter bees. Interestingly, black sage and white sage also prevented from mating by a temporal barrier. Black sage flowers flower in early spring, white sage flowers in late spring and early summer. Presumably, mechanical isolation prevents the insects from cross-pollinating the two species that should happen to flower at the same time. If mating takes place between two species, their gametes may still not combine. Molecular or chemical differences between species cause gametic isolation, in which the egg and sperm of different species are incompatible. In aquatic animals that release their eggs and sperm into the surrounding water simultaneously, interspecific fertilization is rare, extremely rare. The surface of the egg contains specific proteins that bind only to the complementary molecules on the surface of sperm cells of the same species. A similar type of molecular recognition also occurs between pollen grains and the stigma, so that pollen does not germinate on the stigma of a different plant species. Now on to post-zygotic barriers. Fertilization sometimes occurs between gametes of two closely related species despite the existence of pre-zygotic barriers. When such fertilization happens, post-zygotic barriers that increase the likelihood of reproductive failure come into play. Generally, the embryo of an interspecific hybrid spontaneously aborts. Development, the orderly sequence of events that occurs as an organism grows and matures, is a complex process requiring the precise interaction and coordination of many genes. Apparently, the genes from parents that belong to different species do not interact properly in regulating the mechanisms for normal development. In this case, reproductive isolation occurs by hybrid inviability. Hybrid inviability. For example, in the crosses between different species arises, the embryos die before reaching maturity. Similarly, nearly all hybrids die in the embryonic stage when the eggs of a bullfrog are fertilized artificially with the sperm of a leopard frog. If by chance an interspecific hybrid does live, it may not reproduce for any of several reasons. Hybrid animals may exhibit courtship behaviors incompatible with those of either parental species, and the result will be that they will not mate. More often, hybrid sterility occurs when problems during meiosis cause gametes of an interspecific hybrid to be abnormal. Hybrid sterility is particularly common if the two parental species have different chromosome numbers. For example, a mule is the offspring of a female horse, and a female horse diploid number is 64, and a male donkey, and the donkey has a diploid number which is 62. You can see this picture before you now. This type of union is almost always results in sterile offspring, and the diploid number in this case is 63. And it's because of synapsis and the segregation of the chromosomes that cannot occur properly. And occasionally, a mating between two F1 hybrids produce a second hybrid generation, F2. The F2 hybrid may exhibit hybrid breakdown, the inability of a hybrid to reproduce because of some defect. For example, hybrid breakdown in the F2 generation of a cross between two sunflower species was 80%. In other words, 80% of the F2 generation were defective in some way and could not reproduce successfully. Hybrid breakdown also occurs in the F3 and later generations. Please be sure to see Table 21, that's 20-1, which summarizes the various prezygotic and postzygotic barriers. Now on to speciation. We are now ready to consider how entirely new species may arise from previously existing ones. The evolution of a new species is called speciation, the formation of new species. Alright, let me start that over, excuse me. The evolution of a new species is speciation. So the formation of two species from a single species that occurs when a population becomes reproductively isolated. And that's reproductively isolated from other populations of the species, and the gene pools of the two separated populations begin to diverge in genetic composition. So when a population is sufficiently different from its ancestral species, that there is no genetic exchange between them, we consider the speciation to have occurred. So speciation happens in two ways. It's either allopatric speciation or sympatric speciation. So speciation that occurs when one population becomes geographically separated from the rest of the species and subsequently evolves by natural selection and or genetic drift is known as allopatric speciation. So I'll say again, a geographic barrier separates the population in allopatric speciation. So there will be in two separate geographic regions or areas. So if you think back to chapter 18, natural selection occurs as individuals that possess favorable adaptations to the environment survive and become parents of the next generation. And keep also in mind that genetic drift is the random change in low frequencies resulting from the effects of chance on the survival and the reproductive success of individuals in small breeding populations. So in allopatric speciation, it is the most common method of speciation and accounts for almost all evolution of new animal species. The geographic isolation required for allopatric speciation may occur in several ways. Earth's surface is in a constant state of change. Such change includes rivers shifting their courses, glaciers migrating, mountain ranges forming, land bridges forming that separate previously united aquatic populations, and large lakes diminishing into several smaller geographically separated pools. What may bring an imposing barrier to one species may be no consequence to another. Birds and cattails, for example, do not become isolated when a lake subsides into smaller pools. Birds easily fly from one pool to another, and cattails disperse their pollens and fruit by air currents. Fishes, in contrast, usually cannot cross land barriers between the pools, and so become reproductively isolated. In Death Valley region of California and Nevada, large interconnected lakes formed during wetter climates of the last ice age. These lakes were populated by one or several species of pupfishes. Over time, the climate became drier. The large lakes dried up, leaving isolated pools. Presumably, each pool contained a small population of pupfish that gradually diverged from the common ancestral species by genetic drift and natural selection in response to habitat difference such as high temperatures, high salt concentrations, and low oxygen levels, characteristics of desert springs. Today there are 20 or so distinct species and subspecies and populations of pupfish in the area of Death Valley, California, and Ash Meadows, Nevada, and that's what you're seeing here in the figure A and figure B. So many of the Devil's Hoe pupfish and the Owens pupfish are restricted to one or two isolated springs. The Devil's Hoe pupfish is in A, and at B is the Ash Meadows pupfish. Allopatric speciation also occurs when a small population migrates or is dispersed and colonizes a new area from the original species. So microevolutionary forces that change and accumulate in the isolated gene pool over many generations may eventually be sufficient to form a new species. So I won't go through the Hawaiian geese example, but this is also another example of allopatric speciation. So there are a number of examples your text provides, but for the sake of time, I'll move on to what's next. Sympatric speciation. So although geographic isolation is an important factor in the cases of evolution, it is not an absolute requirement. In sympatric speciation, a new species evolves within the same geographic region as the parental species. So to say it another way, by way of sympatric speciation, speciation occurs in that one geographic region where that population is in one area and there is absolutely no geographic barrier involved. The divergence of two populations with the same geographic range occurs when reproductive isolating mechanisms evolve at the start of the speciation process. Sympatric speciation is especially common in plants. The role of sympatric speciation in animal evolution is probably much less important than allopatric speciation. Until recently, sympatric speciation in animals has been difficult to demonstrate in nature. Sympatric speciation occurs in at least two ways: the change in ploidy, which is the number of chromosome sets making up an organism's genome, and the change in ecology. As a result of reproductive isolating mechanisms, as discussed earlier, the union of two gametes from different species rarely forms viable offsprings. If offspring are produced, they are usually sterile. Synapsis cannot occur in interspecific hybrid offspring because not all the chromosomes are homologous. However, if the chromosome number doubles before meiosis, homologous chromosomes can undergo synapsis. Although not common, this spontaneous doubling of chromosomes has been documented in a variety of plants and a few animals. It produces nuclei with multiple sets of chromosomes. Polyploids, which possess more than two sets of chromosomes, have played a major role in plant evolution. Reproductive isolation occurs in a single generation when a polyploid species with multiple sets of chromosomes arises from diploid parents. There are two kinds of polyploid: autopolyploid and allopolyploid. An autopolyploid contains multiple sets of chromosomes from a single species. Autopolyploid, multiple sets of chromosomes from a single species. An allopolyploid contains multiple sets of chromosomes from two or more species. So allopolyploid, two or more species; autopolyploid, multiple sets of chromosomes from a single species. So we'll discuss only allopolyploidy because it is much more common in nature than autopolyploidy. Allopolyploidy occurs in conjunction with hybridization, which is sexual reproduction between individuals from closely related species. Allopolyploidy produces fertile interspecific hybrids because the polyploid condition provides the homologous chromosome pairs necessary for synapsis during meiosis. As a result, gametes may be viable, and allopolyploid, that is, it is an interspecific hybrid produced by allopolyploidy, and it reproduces with itself, so self-fertilization, or with a similar individual. However, allopolyploids are reproductively isolated from both parents because their gametes have a different number of chromosomes from those of either parent. So if a population of allopolyploids becomes established, the selective pressures cause one of three outcomes. First, a new species may not compete successfully against species that are already established, so it becomes extinct. Second, the allopolyploid individuals may assume a new role in the environment and so exist and coexist with both parental species. Or thirdly, the new species may successfully compete with either both of its parental species if it has a combination of traits that confers greater fitness than one or both of the parental species for all or part of the original range of the parents, and then the hybrid species may replace the parents. Although allopolyploidy is extremely rare in animals, it is significant in the evolution of flowering plant species. As many as 80% of all flowering plants are polyploids, and most of those are allopolyploids. Moreover, allopolyploidy provides a mechanism for extremely rapid speciation. A single generation is all that is needed to form a new reproductively isolated species. So allopolyploidy may explain the rapid appearance of many flowering plant species in the fossil record and their remarkable diversity, with at least 300,000, I'll say with over 300,000 species of angiosperms, which are the flowering plants. And what's typical is that polyploids are typically larger as well. So there we have it. And you're seeing here the Q primrose in picture here. So it is an example of sympatric speciation that was documented at the Royal Botanic Gardens at Kew, England in 1898. So plant breeders developed the species as an interspecific hybrid from two primrose species. So they each had 18 chromosomes in their diploid chromosomes. Then the chromosome number of 18 was sterile. Then, at 3 different times, it was reported to have spontaneously formed a fertile branch, which was an allopolyploid, and the diploid number was then 36. It produced viable seeds. So the mechanism of sympatric speciation may be experimentally verified for many plants. One example is a group of hemp nettles that occur in temperate parts of Europe and Asia. One hemp nettle is naturally occurring, allopolyploid. Its diploid number is 32, to are formed by the hybridization of two species, with 16 chromosomes in their diploid, respectively. This process occurred in nature but was experimentally produced when they were crossed. So the hybrids, most of which were sterile. Nevertheless, the F2 and F3 generations produced, and the F3 generation included a polyploid plant with 32 chromosomes, and that is that diploid number, and it self-fertilized to yield an F4 generation that could not mate with either of those parental species. These allopolyploid plants had the same appearance and chromosome number that occurred naturally. So sympatric speciation in animals. So biologists have observed the occurrence of sympatric speciation in animals, but its significance, how often it happens, and under what conditions is still debated. I'll be brief. Many examples of sympatric speciation in animals involve parasitic insects and rely on the genetic mechanisms other than polyploidy. So in the 1860s, in the Hudson River Valley of New York, a population of fruit maggot flies, parasitic on the small red fruits of the native hawthorn trees, was documented to have switched to a new host, domestic apples, which had been introduced from Europe. Although the sister populations, meaning the hawthorn maggot flies and the apple maggot flies, continue to occupy the same geographic area, no gene flow occurs between them because they eat, mate, and lay eggs on different hosts. In other words, because the hawthorn and apple maggot flies have diverged and are reproductively isolated from each other, they have effectively become separate species. Most entomologists, however, still recognize hawthorn and maggot flies as a single species because their appearance is virtually identical. In situations such as that of the fruit maggot flies, a mutation arises in an individual and spreads to a small group of insects by sexual reproduction. The particular mutation leads to disruptive selection, in which both old and new phenotypes are favored. Selection is disruptive because the original population is still favored when it parasitizes the original host, and the mutants are favored by a new ecological opportunity, which in this case is to parasitize a different host. Additional mutations may occur that causes sister populations to diverge even further. And there you see it. Hawthorn maggot fly range is in green, and the apple fly maggot range and hawthorn fly maggot range is there with the hash. Sexual selection. So biologists have studied speciation of the colorful fishes known as cichlids. So this is in several eastern African lakes. So the different species of cichlids in a given lake have remarkably different eating habits, which are reflected in the shapes of their jaws and teeth. Some graze on algae, some consume dead organic material at the bottom of the lake, and others are predatory and eat plankton. Insect larvae, the scales of the fish, or even other cichlid species. In some cichlids, food preferences are related to size. So smaller cichlids consume plankton, which is in turn related to mating preference, and then small plankton-eating cichlids mate only with small plankton-eating cichlids. In other cichlids, related species do not differ in size, only in color and mating habits. Each species has a distinct coloration which matches the preference of females of that species in choosing mates. Darwin called choosing a mate based on color or some characteristic sexual selection. He recognized the female preference for showy colors and elaborate ornaments on males. If it was based on inherited variation, could result in the male sexual traits becoming more common and more pronounced over time. Female preference for a particular male trait confers a selective advantage to the males exhibiting that trait that they have improved the chance to mate and pass on their genes. Darwin suggested that sexual selection, like natural selection, could eventually result in the evolution of new species. So DNA sequence data indicate that cichlid species within a single lake are more closely related to one another than to fishes in nearby lakes or rivers. These molecular data suggest that cichlid species evolved sympatrically, or at least within the confines of a lake, rather than by repeat colonization by fish populations in nearby rivers. So how rapidly did sympatric speciation occur in cichlids? So seismic and drill core data provided evidence that Lake Victoria dried up completely during the last Pleistocene, when much of northern and equatorial Africa was arid. The diverse cichlid species may have risen after the climate became wetter and Lake Victoria refilled. Further, the more than 500 endemic species, an endemic species is a species that is found nowhere else on Earth, of cichlids in Lake Victoria have evolved from one or a few common ancestors in a remarkably short period of time, as little as 12,000 years. Other biologists have offered competing hypotheses about the timing and the geographic origin of Lake Victoria's cichlids. Regardless of the uncertainties of the details, Lake Victoria's cichlids are widely recognized as an exemplifyingly fastest rate of evolution known for such a large group of vertebrate species. Let's move on from these cichlids. So I won't get to hybrid zones, which a hybrid zone is an area of overlap between two recently diverged populations in which interbreeding takes place and hybrid offspring are common. And hybrid zones are typically narrow, presumably because hybrids are not well adapted for either parental environment. And the hybrid population is typically very small compared to the parental populations. Let's get over to what's next. The rate of evolutionary change. So two different models, punctuated equilibrium and phyletic gradualism, explain evolution as observed in the fossil record. So punctuated equilibrium model was proposed by paleontologists who questioned whether the fossil record is actually incomplete as it initially appeared. And in the history of species, long periods of stasis appeared to be punctuated and interrupted by short periods of rapid speciation that were perhaps triggered by changes in the evolutionary environment. That is, periods of great evolutionary stress, and speciation therefore proceeded in spurts. So punctuated equilibrium accounts for the abrupt appearance of new species in the fossil record with little to no evidence of intermediate forms. So in contrast, the traditional Darwinian view of evolution espouses phyletic gradualism, in which evolution proceeded continuously over long periods of time. So gradualism is rarely observed in the fossil record because the record is incomplete. So occasionally a complete fossil of a transitional form has been discovered and cited as a strong case for populations slowly diverge from one another by the gradual accumulation of adaptive characteristics within each population. So these adaptive characteristics accumulate as a result of different selective pressures encountered in different environments. So here you're seeing the key point. This is figure 2015, showing the difference between punctuated equilibrium. So you see punctuated equilibrium here at A. Long periods of stasis are interrupted by short periods of rapid speciation. And then in B, you're seeing phyletic gradualism. So a slow, steady change in species occurs over time. Macroevolution. So macroevolution concerns large-scale phenotypic changes in populations that generally warrant their placement in taxonomic groups at the species level and higher. That is, new species, genera, families, orders, classes, and even new phyla, kingdoms, and domains. One concern of macroevolution is to explain evolutionary novelties, which are large phenotypic changes, such as the appearance of jointed limbs during the evolution of arthropods, crustaceans, insects, and spiders. These phenotypic changes are so great that the new species possessing them are assigned to different genera or higher taxonomic categories. So studies of macroevolution also seek to discover and explain major changes in species diversity through time, such as occur during adaptive radiation, when many species appear, and mass extinction, when many species disappear. Thus, evolutionary novelty such as adaptive radiation and mass extinction are important aspects of macroevolution. Adaptive radiation. Some ancestral species have given rise to far more species than have other evolutionary lineages. Adaptive radiation is the evolutionary diversification of many related species from one or a few ancestral species in a relatively short period. Evolutionary innovations, such as those we considered in the last section, may trigger an adaptive radiation. Alternatively, adaptive radiation may occur because the ancestral species happens to have been in the right place at the right time to explore numerous ecological opportunities. So biologists developed the concept of adaptive zones to try to help explain this type of adaptive radiation. And an adaptive zone is a new ecological opportunity that was not exploited by the ancestral organism. So at the species level, an adaptive zone is essentially identical to one that is similar ecological niches. Examples of adaptive zones includes nocturnal flying to catch small insects, grazing on grass while migrating across the savanna, or swimming in an ocean's surface to filter plankton. When many adaptive zones are empty, as they were in Lake Victoria when it was refilled some 12,000 years ago, colonizing species such as the cichlids may rapidly diversify to exploit them. And because islands have fewer species than do mainland areas of similar size, latitude, topography, vacant adaptive zones are more common on islands than on continents. Consider the Hawaiian honey creepers, as you'll see here. A group of related birds found on the Hawaiian Islands. When the honey creeper ancestors reached Hawaii, few other birds were present. The succeeding generations of honey creepers quickly diversified into many new species, and in the process occupied the many available adaptive zones that on the mainland are occupied by finches, honey eaters, tree creepers, and woodpeckers. The diversity of honey creeper bills is a particularly good illustration of adaptive radiation. Some honey creeper bills are curved to extract nectar out of tubular flowers, whereas others are short and thick, ripping away bark in the search of insects. Another example of a vacant adaptive zone includes the Hawaiian silverwarts, which I won't get to the silverwort species of plant, but I will say this is a great example of adaptive radiation, meaning the evolutionary diversification of many related species from one or a few ancestral species over a relatively short period of time. Now on to extinction. I'll be very brief. It is the permanent end of a lineage. In other words, it's when the last individual of a species dies. So it's estimated that only one species is alive today for every 2,000 that have become extinct. And extinction is the eventual fate. It is the eventual fate of all species. So this has been Chapter 20. Please prepare yourself well for your test. And please also look at the summary focus at the end of the chapter. It will help to summarize a lot of what you've been given. This has been your instructor, Scholar Huff. Thank you all for listening.